Hi friends, hope you are fine. At the end of this video, you will be able to understand major enzymes used in molecular biology, different classes of enzymes used in molecular biology laboratory, how it works with examples. Please watch till the end for complete understanding. Let's begin with the classes of enzymes in molecular biology. The first one is the nucleases. Nucleases are nucleic acid degrading enzymes. It includes endonuclease, exonuclease, ribonuclease, and DNase I. The second class is called as DNA modifiers that include DNA polymerase, reverse transcriptase, alkaline phosphatase, polynucleotide kinase, terminal nucleotidyl transferase, and methyl transferase. And finally, the ligases, DNA ligase and RNA ligase. Starting with nucleases. Nucleases are nucleic acid degrading enzymes. So there are two types of nucleases. Suppose this is a DNA fragment. Two classes of nucleases are endonucleases and exonucleases. As the name suggests, endonucleases cuts within the DNA molecule, whereas exonucleases cuts at the ends of the DNA molecule. External. Restriction endonuclease is widely used in gene cloning procedure. So nucleases cleaves phosphodiester bonds in a nucleic acid backbone. They are nucleic acid hydrolyzing enzymes, belongs to two classes. There are two types, endonuclease that makes cuts within a DNA molecule, whereas exonuclease removes nucleotides from the ends. Now let us see the common nucleases that is used in a molecular biology laboratory. Nuclease S1, zinc ion is required for its activity. It degrades RNA or single-stranded DNA into 5' mononucleotides. It is used in removing single-stranded protruding ends of a DNA or single-stranded overhangs of a DNA. The second one is exonuclease. Third, manganese or magnesium ions are required for its activity. 3' exonuclease on double-stranded DNA. It will remove nucleotides from the 3' end, widely used in DNA labeling or DNA length reduction along with nuclease 1. The third one is exonuclease 7. So this enzyme degrades single-stranded DNA from both ends. It has no activity on RNA or DNA-RNA hybrids. It is used in the removal of or protruding ends, also used in the removal of primers from a completed PCR reaction. These are the common enzymes or nucleases that is used in a molecular biology laboratory. Just to remind you, we have given a number of videos on core concepts of biotechnology. You can refer that for more. The link is on the iCard and also in the description. The next nuclease is the ribonuclease. As the term indicates, RNA degrading enzymes, ribonucleic acid degrading enzymes. It cleaves phosphodiester bonds in an RNA backbone. So this is an mRNA with poly A tail. So we'll be making an oligo-DT primer. Using reverse transcriptase, we'll be making a cDNA. Now we have a hybrid with RNA and DNA. So this is an RNA-DNA hybrid, and this ribonuclease is used to remove this mRNA from this RNA-DNA hybrid. Then we can synthesize the second strand to make a double-stranded cDNA using DNA polymerase. So RNase H is an RNA degrading enzyme that selectively removes mRNA from RNA-DNA hybrid to separate the cDNA to synthesize the second strand or to make a double-stranded cDNA. Now let us see the common ribonucleases used in molecular biology laboratory. First one is pancreatic ribonuclease, also called as RNase A, degrades RNA in the 3' end, cleaves RNA in DNA-RNA hybrids at site of single mismatch, so widely used in mutation mapping, also involved in removing RNA from DNA preparation. Then there is ribonuclease H, that is widely used to degrade RNA strand in a DNA-RNA hybrid. So widely used in the removal of RNA probes and also removal of poly A tails of mRNA. And the third one is RNase T1 that cleaves single-stranded RNA at guanine residues. That is widely used in RNA sequencing. And the next one is DNase I, It's a DNA degrading enzyme. So suppose this is a double-stranded DNA. So upon treatment with DNase I, we'll be getting short oligonucleotides or short fragments of DNA. So it's a DNA-specific endonuclease, making internal cuts in a DNA molecule. It hydrolyzes phosphodiester bonds, producing mono or oligodeoxyribonucleotides with 5' phosphate and 3' OH groups. As you see, this is a 5' phosphate and this is a 3' end. DNase I, the requirement is divalent cations like calcium and is activated by magnesium and manganese ions. It cleaves single or double-stranded DNA, may be isolated or even incorporated in a chromatin. Widely used in nick translation of DNA, dideoxy sequencing, elimination of DNA in RNA or protein preparations. So to avoid DNA contamination in RNA or protein preparations, to get pure RNA or pure protein, and also widely used in DNase food printing, which is a technique that is used to find out the protein binding site in a DNA molecule. And the second class is the DNA modifiers. First one is DNA polymerase. It's a DNA-dependent DNA polymerase. Let us understand how it works. This is a double-stranded DNA. After denaturation, then primer is, we will be making a primer that is complementary to the template strand. So we have a forward primer and a reverse primer. All DNA polymerase requires a free 3' OH end for elongating further. In this reaction mix, there will be DNTPs, then there will be buffers with magnesium chloride. DNA polymerase can elongate this strand further from this 3' OH end of the primer. So DNA polymerase is involved in extension or elongation of a strand by nucleotide addition. So this is the new strand. It's a template-directed enzyme that adds complementary nucleotides to the free 3' OH end of the strand. So as you see, this is the newly synthesized strand by DNA polymerase, and this is also the newly synthesized strand by DNA polymerase. So DNA polymerase is an enzyme that adds nucleotides or that elongates a DNA fragment. So this is what is happening. So these are the newly added nucleotides. Common DNA polymerases used in molecular biology includes common prokaryotic DNA polymerases, polymerase I, which requires a template, primer and DNTPs, which has 5' 3' polymerase activity, then 3' 5' exonuclease activity, that is called as a proofreading activity, to remove any incorrectly added nucleotides from the growing strand. Then there is 5' 3' exonuclease activity. So this enzyme is widely used in DNA replication, nick translation, removal of 3' overhangs from DNA ends, and also involved in primer removal. And polymerase II, the requirement is same as polymerase I. It has 5' 3' polymerase activity, then also 3' 5' exonuclease activity. It's widely used in DNA replication. Then polymerase III, also the same requirement with the same feature as of DNA polymerase II, widely used in DNA replication. And there are thermostable DNA polymerases that is primarily used in polymerase chain reaction as the temperature goes up to 72 degrees Celsius. So these thermostable DNA polymerases are isolated from thermophilic bacterium. The most common one is Taq polymerase. It requires a template and also magnesium chloride. Optimal temperature is 80 degrees Celsius. It has 5' 3' polymerase activity and 5' 3' exonuclease activity. So there is no proofreading activity, widely used in PCR reaction, but without proofreading activity, therefore low fidelity or low precision. Now we have PFU, and when these are thermostable DNA polymerases with proofreading activity, it has the activity of Taq plus 3' 5' proofreading activity or exonuclease activity that makes it a thermostable DNA polymerase with high fidelity, widely used in PCR for making of new strands. So these are the common DNA polymerases used in a molecular biology laboratory. Now the next one is reverse transcriptase. It's an RNA-dependent DNA polymerase. We isolated this reverse transcriptase first from viruses. retroviruses. So this enzymes makes DNA strand using mRNA as template strand. So it is used in making of cDNA from mRNA for cDNA library preparation. Let us see how this works. So in cDNA synthesis, first we have an mRNA. mRNA is having a poly A tail, so it's very easy to design a primer. Oligo-DT will work as a primer. Then we'll be using this enzyme reverse transcriptase, and reverse transcriptase use this mRNA as a template for the synthesis of a DNA, cDNA. So this enzyme catalyzes the formation of cDNA from this mRNA template. That's why it is called as RNA dependent DNA polymerase. Then what we do is we have newly added DNA using RNA template. Then this ribonuclease H is used to remove this mRNA we have already discussed. Then DNA polymerase will make the second strand and finally making a double-stranded cDNA that is widely used in the making of cDNA library preparation. So we have given a video on steps in the construction of cDNA library. You can refer that for more. Common reverse transcriptase used in molecular biology laboratory includes AMV-MAV reverse transcriptase that requires RNA template and a primer. Activity is reverse transcription activity, also has RNase H activity, widely used in the synthesis of cDNA from RNA. Then MULV reverse transcriptase that also requires an RNA template and a primer. It has the same activity as that of AMV reverse transcriptase, widely used in RT-PCR for long transcripts. Then there are thermostable reverse transcriptases like MonsterScript RT that requires a template and a primer. It has reverse transcription activity. It can make a cDNA from an mRNA template, but no RNase H activity, widely used in RT-PCR for making long transcripts. And the next DNA modifier is alkaline phosphatase. Alkaline phosphatase removes terminal phosphate groups at the 5' end of a DNA, thus prevents self-annealing of a vector DNA while preparing a vector in gene cloning procedure. So let us understand how this works. So this is a DNA strand. This is the 5' end with the phosphate. Here also in the second strand, 5' end with the phosphate. When we treat this with alkaline phosphatase, phosphate is removed by alkaline phosphatase. Let me make it more clear. So this is a nucleotide. So this is a sugar, nitrogenous base, and this is the phosphate, 5' phosphate. So in the presence of water, alkaline phosphatase can cause a hydrolytic cleavage. That means in the presence of water, it can remove this phosphate. So this phosphate is removed, and this is how alkaline phosphatase works. Removal of terminal phosphate at the 5' end of a DNA. The advantage is in the making of a vector, if there are open ends, there is a chance of re-annealing of vector or self-annealing of vector. So this can be avoided, as without this phosphate there won't be any phosphodiester formation or any re-annealing of a vector DNA, and this phosphate is removed. So widely used in dephosphorylation of DNA or removal of phosphate from a DNA. And the next DNA modifier is polynucleotide kinase. Polynucleotide kinase transfers or add phosphate from ATP to 5' OH group of a dephosphorylated DNA or RNA. So the last enzyme, alkaline phosphatase, removes phosphate from a DNA, whereas kinase enzyme adds phosphate to a DNA fragment. Let us see how this works. So this is a DNA strand. From this ATP, the phosphate is added. So 5' phosphate is added by this polynucleotide kinase. ATP becomes ADP and the phosphate is transferred to this 5' end. So kinase is involved in adding phosphate from ATP to the 5' OH group of a dephosphorylated DNA. So it's widely used in rephosphorylating a dephosphorylated DNA in the case of vector RNA or DNA, widely used in radiolabeling to transfer radioactive phosphorus 32 from ATP to dephosphorylated 5' end of DNA or RNA. First we'll be removing the phosphate using alkaline phosphatase, then we'll be transferring a radioactively labeled phosphate using this polynucleotide kinase. Thus, this enzyme is widely used in radiolabeling. Let me summarize these two enzymes. So these enzymes are involved in phosphate removal and transfer. Alkaline phosphatase requirement is zinc and magnesium ions, removes 5' phosphate groups from nucleic acids to prevent re-annealing or recircularization of DNA vectors in gene cloning procedures, whereas polynucleotide kinase that requires ATP and magnesium ions transfers a phosphate group from ATP to 5' OH end of a nucleic acid. Then it is involved in nucleic acid labeling. And the next DNA modifier is terminal nucleotidal transferase. This enzyme adds nucleotides to the 3' OH group of a DNA fragment, used to make homopolymer sticky or cohesive tails at 3' end of a DNA fragment, helps in joining fragments with blunt ends, making it sticky. Also used to make radioactive DNA probes by end labeling. Let us see how this works. So this is a double-stranded DNA molecule. In the presence of this enzyme, terminal nucleotidyl transferase, it helps in poly-A tailing. As you see, poly-A tailing is done. This is called as homopolymer tailing. So this will produce a single-stranded overhangs or sticky ends that is widely preferred in gene cloning procedure. Any fragment with complementary strand can anneal easily by formation of hydrogen bonds. That is why sticky end is preferred in gene cloning procedures. And same is the case with this fragment also. We can add poly-T tail also. So this terminal nucleotidyl transferase, which is widely used in homopolymer tailing to make a blunt and sticky, so that that fragment can be easily used to anneal another fragment. So terminal nucleotidyl transferase is widely used in homopolymer tailing, making a blunted fragment sticky by the addition of poly-A tail or poly-T tail. Next DNA modifier is methyl transferase. Methyl transferase enzyme that adds a methyl group to cysteine or adenine residues of a DNA. It's used to methylate desired DNA in the recombinant DNA to protect it from cleavage by restriction enzymes or the restriction systems of the host. Let us see how this works. As we know, EcoR1, a restriction enzyme, a restriction endonuclease, its recognition sequence is GAATTC. So this is a recognition sequence. So whenever this sequence is encountered by this enzyme, it can make a cleave and forms fragments like this, forming sticky ends. So this is how this enzyme makes cuts. If this segment or this fragment is methylated, if this, in the presence of methyl transferase, if this adenine residue is methylated, then this enzyme cannot cleave this fragment as this fragment is methylated. So methylation offers protection from digestion by restriction enzymes and also helps in protection from cleavage by restriction enzymes of the host. Even in bacteria, methylation is a method by which bacteria protects its own DNA from cleavage by restriction modification system. Foreign DNA may not be methylated, so it will be cleaved, whereas bacteria's own DNA is methylated so that it is protected from these restriction enzymes. Hope you are clear. So there is no cleavage by EcoR1. And finally, ligases. Ligases are joining enzymes. Suppose this is a DNA fragment 1 and this is a DNA fragment 2. These two DNA fragments are joined by ligases by the formation of phosphodiester bond. So ligases are joining enzymes, or also called as molecular glue, that joins DNA fragments by forming phosphodiester bond. The process is called as ligation. So widely used to join DNA fragments with blunt or sticky ends, add linkers or adapters to DNA, or to repair nicks. So has wide application. It's a sealing enzyme. So this is how DNA ligases works. This is called the nick. A phosphodiester bond is missing. So ligases catalyzes the formation of phosphodiester bond between this 3' OH end and 5' phosphate group at this single-stranded break in a double-stranded DNA molecule. It requires ATP or NAD+ for its activity. Common example is T4 ligase. Then ligases commonly used in molecular biology laboratory includes T4 DNA ligase that requires magnesium ions and ATP. It connects blunt end and cohesive ends in double-stranded DNA, RNA or double-stranded RNA hybrids, widely used for cloning or gene cloning procedures. Then there is E. coli DNA ligase that requires magnesium ion. It connects preferentially cohesive double-stranded DNA ends in the presence of phycol or PEG polyethylene glycol, widely used in ligation when blunt-end RNA or DNA ligation needs to be avoided. Then there are RNA ligases. T4 RNA ligase 1 that requires ATP for its activity. It ligates single-stranded nucleic acids and polynucleotides to RNA molecules, widely used in RNA labeling and primer extension. Then there is T4 RNA ligase 2 that also requires ATP. It ligates double-stranded RNA or connects double-stranded RNA to double-stranded DNA. It repairs nicks in double-stranded RNA. So these are the common enzymes used in molecular biology laboratory. Let me summarize. The three classes. First one is nucleases or nucleic acid degrading enzymes that include endonuclease that makes internal cuts in a DNA molecule. Then there is exonuclease that makes cuts from the ends of a DNA molecule. Ribonucleases are RNA degrading enzymes, and DNase I is also a DNA degrading enzyme. DNA modifiers includes DNA polymerase to make new strands by the addition of nucleotides, then reverse transcriptase, that is an RNA dependent DNA polymerase that makes a DNA strand from an RNA template, then alkaline phosphatase that is used to remove a phosphate group, whereas polynucleotide kinase is used to add a phosphate group, whereas terminal nucleotidyl transferase is used in homopolymer tailing, poly-A tail or poly-T tail, and finally methyl transferase adds methyl residues to adenine or cysteine residues that helps in protection from restriction digestion by restriction enzymes. And finally, ligases are enzymes that ligates or joins two DNA fragments. The common ligases are DNA ligases and RNA ligases that joins two fragments by the formation of phosphodiester bonds. Hope you are benefited from this video. If you find this video useful, please consider subscribing our channel. Take care. Stay blessed. Thank you so much for your support. You are with biologyexamsforyou.com.